Hello and welcome everyone to CyVerse's Focus Forum webinar series. I'm Tina Lee, CyVerse's User Engagement Officer. Today we are pleased to present a webinar entitled Doing Metagenomics Using QIIME 2 with Jupyter Notebooks in CyVerse Vice, presented by Jocelyn Lee, a Science Education Fellow at Howard Hughes Medical Institute. She's also a CyVerse staff alumna. I'm going to do some quick housekeeping and then turn the webinar over to Jocelyn shortly. So first of all, please open your chat windows where you can type in questions for Jocelyn to address after her presentation. Today's presentation is roughly 30 minutes, and we will again address the questions at the end. Materials from today's webinar, such as Jocelyn's slides and a YouTube video, will be posted on a wiki page that you can visit at any time to replay your favorite parts of today. Also, I'd like to let you know about some upcoming trainings that CyVerse is offering. It's called the Foundational Open Science Skills Camp. We call it FOSS. It's a camp-style event in June at the U of A here, and it's intended to give new PIs and early career researchers a solid foundation in free open source computational resources and open science skills in order to implement their projects more effectively. Also, our next webinar, called Nanopore Sequencing Data Analysis in CyVerse Discovery Environment, will be presented in two parts by Upender Devasetti and Amanda Cooksey on April 19th and May 3rd, respectively. Please go to our events page on our website for more information and to register. After today's webinar, we'll also email you the links to the wiki page with the information and the links, and links to our website learning page for information on FOSSCamp, and with a brief survey to get your feedback. If you have ideas for future webinars, please feel free to send them to me in the chat window and we'll do our best to arrange something. So without further ado, please meet today's presenter, Jocelyn Lee, whose research interests include microbiome research, enzyme catalysts, and bioinformatics education. We're pleased to have her and lucky to have her as a presenter today. So welcome, Jocelyn.
Thank you, Tina. That was a great overview of all the upcoming events, and I really enjoyed my time with CyVerse, so I'm glad to share and continue collaborating with CyVerse in the future. So good afternoon, welcome everyone. I'm going to start with the slides, and again, these will be available at the end of the presentation. Let's see. So for today, I wanted to highlight what areas I plan to cover. I'll give a brief introduction of CyVerse to those who may not be too familiar with this infrastructure, and then talk about some of the updates for QIIME 2, and then sort of cross over into how you can run QIIME 2 on CyVerse through the Discovery Environment's new Vice area. Also highlight that it's important to be able to reproduce your data, so using Jupyter Notebooks in complement to run QIIME 2 is a very useful way to check back what you were doing 6 months ago. So I'll go ahead and demo at the end of this how you'll be able to log into the discovery environment and be able to check out some Jupyter Notebooks I've created, and then we'll go over a short demo. I want to also highlight that due to time constraint, I'm not going to cover a full tutorial of QIIME 2. If that's a very big interest with participants, I can do something along those lines as a YouTube video at a later time. So what is CyVerse? It has a vision of transforming science through data-driven discovery and a mission to design, deploy, and expand a national cyber infrastructure for life sciences research and to train scientists in its use. So this is a NSF-funded project that is freely accessible to individuals in the US, and you get to have a place to host your data. So one of the main components of CyVerse is our data store and how it's connected to various products, high performance computing, like Exceed, the computational grid of Open Science, and our cloud computing of Atmosphere. But the way you can interact with all of this is through a multi-client access. So the command line, a web client, or a desktop client. I guess what I use most commonly for that is CyberDuck. So they're all different components, and there's some videos that you can check out a little bit more to know about CyVerse. So one of the products that I highlighted was the discovery environment, and this is at the website de.cyverse.org. Initially, when this was developed a few years ago, it was how can we get command line applications to run in a more graphical user interface. So when you're launching these larger jobs and you're not too savvy with the command line, it's one way to at least get you to analyze some data. So the exciting part of this is it's complemented with our high performance computing. And most of these, if you're familiar with adding apps to CyVerse, it's through using Docker. And if this doesn't make so much sense and you're more on the user side, that's okay for this. So I want to highlight a little bit of this new addition to the discovery environment, which is the Visual and Interactive Computing Environment, VICE. And there is a previous webinar on this back last year. So if you're interested to know more of the ins and outs of VICE, you're more than welcome to check that out. So what VICE does is you're able to launch web applications around Docker. So how we've gotten QIIME 2 to run in the discovery environment is through a Docker container. So this gives you an easy way to access through a secure and authenticated URL. And this will make a little bit more sense once we move into the demo portion. So now I'm going to switch from the infrastructure, so that's where we run all the computational analysis, and sort of take a step back. So re-highlight where in a microbiome study, QIIME 2 comes into this area. So for those of you who may be on the study design side, you go through and collect your samples, collect metadata. You send that genomic DNA to your sequencing center. They or you may be adding the barcodes or adapters for sequencing. So then they'll sequence either your amplicon sequencing or the ITS region, so whichever your microbiome sequencing is doing, you'll get your output reads. And this is where the data processing comes into how do you find out what's in your sequence samples. So in this schematic here, I'm showing that the downstream part for your data processing and the data analysis is where you can use tools like QIIME 2. So QIIME 2 stands for Quantitative Insights into Microbial Ecology. It's pronounced QIIME. It's what the developers call a decentralized microbiome analysis package. So it has a focus with analysis transparency and really being able to track your data along the way. For this, QIIME 2 provides a way that you can start with raw DNA sequence data and finish with some figures and statistics. Those of you familiar with other types of tools like Mother, or when you use standalone denoising and clustering software like Deblur or Data2, those are all written by developers. What QIIME 2 does is it wraps it into a nice start to finish workflow. And there are plugins involved with this that can do each of those steps. So QIIME 2 has a few features where you can generate visualization files, but really focus on the bioinformatic methods that the general community uses. One thing I've really liked is all the documentation that's been available. You get a few of your moving pictures or the Atacama Desert data sets to use. So those are a nice way to start when you're first learning about how to run through QIIME 2. And one thing I've found being a part of the QIIME 2 community is that the bioinformaticians and developers are all very supportive to help you learn in this area. One really exciting part of QIIME 2 I'm wanting to highlight is the provenance tracking. So this is a way that you're able to run one of the plugins for QIIME 2 and all the parameters, and when you ran this, all are recorded during this process. So it's really exciting. And if you have a tool that you want to integrate into the QIIME community, there's ways to have that fit into the plugin architecture so it's accessible to users for things. When we think of reproducibility, you want to identify what version of a program you did or used. You have to understand what input files you are using, all the metadata associated with your sample collection. So all of this needs to be recorded somewhere. So that's one thing you have to keep in the back of your mind once you're going through this data analysis. And you know, I'm showing an example here of a data set that was recently published. So it's doable as long as whoever's publishing the data set gives you all that information in the program. So this is a quick screenshot right here on the left. I'm showing a visualization file through QIIME, and this is a taxa bar plot. So you have samples all listed in these individual bars, and they break down into what was taxonomically assigned for this. If you look up at the top within this data zipped file, there's this provenance, which will show you what actually occurred to your step. So at the bottom of this is a square that is what an action was done to. So you had a feature table and you performed a taxonomy for this. So the output for this taxa bar plot is the end level. If you go up a step to see where all of that came from, is this what I clicked on, a green-gray color, and that's where we did the demultiplex sequences to cluster and denoise. And if you look in the provenance action details, it'll tell you your method and that you used a blur. So this will make a little bit more sense once we go into it, but it's really exciting that you can also list the parameters, which is helpful because this QZV file, it's a visualization file. It has all of this all put together through this zipped, I guess, QIIME file format. So this is really exciting that I think the individuals for QIIME have expanded on. So the QIIME viewer, this is also a recent update. So all of the output files that you generate running each step of QIIME are in what we call these zipped QZA artifact files, or these QZV visualization files. So let's say you have a collaborator who doesn't really know the ins and outs of how to install QIIME. You can actually send them these two file types, and they can upload them by going to a QIIME viewer on their internet browser, and they can drag and drop that file into the web browser and be able to visualize that taxa bar plot or some of the alpha and beta diversity. So it's really exciting if you have individuals who really don't want to install QIIME on their computer. One other space that I think is very supportive is a forum. So if you have issues trying to import your data, or you have a format that you're not sure how to work with, a lot of the technical support is very user-friendly. So it all depends on, if you have general discussions, this is one area to start before you get frustrated. And it's a very supportive community of moderators who also help you do your best to work with your data. So now that we know the infrastructure, we have the bioinformatics tool we want to analyze our amplicon sequence data. How do you take notes of that? Some folks usually use like a text editor, but a really awesome notebook is called Jupyter Notebook. So if you haven't heard of this, I would recommend that you begin to think of how you record all the steps on your bioinformatics workflow. And this is all part of Project Jupyter, so I would recommend you to check this out. So Jupyter is for Julia, the J-U, P-U-I for Python, and the R at the end for R. So these were the first types of notebooks that you would be able to code and write in those programming, or those languages, I guess is the best way to put it. And also they've expanded, so it's not just Julia, Python, and R. You can begin to explore a little bit more on what we have for Vice. So I tried to get through this because I think the more exciting part is showing a live demo. So I'm going to get out of PowerPoint now and then move into showing you a little bit around CyVerse.org so you're familiar with things. So let me exit out. And I'm going to click this link, open. All right. So if you are following on the PowerPoint a little bit later and you click on that link, that'll take you directly to our documentation for advice. So this is where you can learn a little bit more about how to begin using Jupyter Lab or our studio apps, but I'm going to take a step back because I forgot I need to show you CyVerse. So if you go to type in CyVerse.org, this will take you to our home site. And those of you that are already registered users, you can go to your account, and if you need to sign up for a free account, you can use a .edu address to be able to work with our services. So this is what we call our user portal, and where advice is listed is under discovery environment, so we can launch it here if you're just starting at CyVerse.org. But if you're in this venue, we have, those of you that are users that are familiar, we have 3 buttons that are our data, apps, and analysis. And this is sort of our home graphical user interface that we're able to utilize different types of bioinformatics tools. So one way you can go to find out how to launch QIIME 2, that is one of our apps. So you click on the apps icon. And you can search through this with Jupyter, Jupy, and this will take a little bit to load all the possible names. And the most current one we have is Jupyter Lab QIIME 2 2018.11. So this version, we're trying to keep up on how often QIIME 2 is updated on their side. So if you really need the latest version, you can work with me or Upendra for identifying when we need to update this. But the most stable release that I've used is the 0.11. But we'll keep folks updated and try to do our best to have the most current version. So this box is what we call the app, and you can hover over the title and click on this, and this will or present a new window. And this is where we need to select parameters for our app to run. So you can give it an analysis name. I'm going to put it as today. So let's see, analysis. I'm going to keep lower cases. 2019-04-05. I like to leave myself information, so I will type in demo for focus forum. It's always good to get used to, leaving yourself some tips on how did I get here. And then I'm going to have all the output that I generate during my time running this analysis to be in the analysis folder. So this is our default for things. For parameters, I'm going to, go ahead and select a folder. So this is where you'll be able to connect to your data store. So my CyVerse username is Jocelyn, just to keep it simple. I'm gonna, navigate to the community data. So all of the demo data sets, so this is a new data set that I decided to check out. You always want to reproduce someone else's sample. Data that they published on. So this community data is a folder with shared resources. So I chose a acute leukemia lymphoma data set that was a publication from February of this year, and I wanted to run through that data. So I will share this probably on the PowerPoint so that you're able to navigate to example data. So this file system is iPlant Home Shared iPlant Collaborative example data, and it's at the very end of QIIME 2. So let me bear with me as I scroll. We have lots of data, so if you have other analysis to do, this is one thing. So QIIME 2 is where all of this gut microbiome data set is located. So it might take a bit to upload here. And again, this is a public data set, so I downloaded it from the European ENA. That's where they host some of the raw data. I had to go through to figure out how to go ahead and reassign sample IDs. So I did the heavy work for you because it really was all the raw data for this. Okay, so in here, this is a little problematic, but we'll fix it. There were supposed to be a few other files in here, but maybe they'll show up. Let's just do QIIME 2 for now. I have a backup already set up, so we'll see this and we'll make sure that if you want to access this data, it works. So click OK. So the input folder will be all the contents that are in this example QIIME 2 data. So once I have all of these set up, I'm going to click on these arrows up to bring you back to show you this is the input information. And I'm going to click on Launch Analysis. So when you're doing this on your own pace, it'll take probably 5 to 15 minutes for you to have a already secure link to be able to view your Jupyter Lab setup. I want to show one alternate way before we go into that. So I'm going to go back to that Vice documentation. So this is that link that was in the PowerPoint. So Vice, this is if you're interested in knowing a little bit more about this tool. But if you go onto the left side and navigate to list of Vice apps, and you don't remember, what happened, you can go to the Jupyter Lab QIIME 2 name, and this is the name of that same app we just launched previously. But what was really cool is that shows you more details on how to quick start. So say you forgot where to find this YouTube, you're able to look for Vice apps within CyVerse, read the docs, and this is where you can find out the platform that you're using, learn more information about the DE, learn about the input files, that gut microbiome folder that I showed. And it'll also go through, if you don't remember fully, all of the steps to launch your analysis. And if you really don't care for that too much, you can use this quick launch app. So this is something new that Upendra and I had talked about. But again, this will open directly up into the discovery environment and already navigate to your app. So there are multiple ways to do different things on here, just to keep in mind. So, okay. Because again, this is a demo, I want to show a few things. I already have an analysis running, so I'm going to go ahead and connect to that. So once you have your link live and running, you'll get a notification. I got logged out, so I'm just going to re-log into CyVerse using my credentials. So now it's going to pop up and you can see the URL changes, and this is your opening screen of Jupyter Lab. So Jupyter Lab is a way that you're able to view all of your current files within that launched Jupyter instance. And you can see that you have access to notebooks that are in Python, Bash, Julia, or R environments. So this is a cool way that you'll be able to work and connect with your data. So you have various headers of the file, and if you're new to Jupyter Lab and Jupyter Notebook, this might take a while to understand how to navigate, but I do recommend you play around with various items. So, okay, so I'm going to go ahead and the first thing I would recommend is connect to the data store. So this is a way that you're able to transfer data. So whenever I run an analysis, I want to take it from this discovery environment setup and make sure it's backed up somewhere. And you can download directly from the Jupyter Lab, but you also want to be able to save it somewhere else. So this Irods setup configuration already has the inputs that you normally need to connect to the data store. So you enter your CyVerse username. Mine is Jocelyn. And I'm not going to say my password out loud. And I'm going to click Submit. And let me push this up. I will now be able to see all the items within my CyVerse data store account. So if you're a newer user, you're not going to have all these files because I've been running many things on here. What I want to do next is I can't really drag and drop large files or folders, so I need to connect to the data store into a terminal so that I can, once I launch my Jupyter notebooks, I'll be able to work with them. So if you're new to iCommands, I didn't post any information on that just yet, but you have to initialize. In order for the app to connect to the data store, you have to set up this configuration, open up a terminal, and type in IINIT, and this is initialize. I don't know if I can make this a little bigger. Let's do that. Okay. So IINIT, Initialize Irods and iCommands. And for those of you who have used the iCommands before, you notice you don't have to input a few of the Irods configuration setup. So I can type in ILS. That's saying list all the contents within the data store, and you'll see that you'll get all the same inputs that we see here on the left. But you can be able to navigate iCommands there. So now that we're connected back to the data store, let's actually get to see what a Jupyter notebook looks like. So I believe they should, okay, all right. So this icon on the left is your home. Once you log into Vice through DE, all of this is gonna be housed in the Vice folder. So a little bit of this file system can be a little bit tricky to get used to, but once you play around, it makes more sense. So when I launched my app, I selected the gut microbiome folder to be loaded released, or I guess at the same time as I was launching the Jupyter notebook. So if I click on gut microbiome, you can see that there are all these files, and these are from the paper. I'm going to scoop this. Oh, can I expand this? So these are all the raw FASTQ files that came from the publication. So I just wanted to show that. And it looks like the Jupyter notebooks were not transferred over. So I'm going to upload from my, well, let's see. I'm going to investigate here. Yeah, ILSFF output. They might be saved on this part of, okay, so there's two of them. Bear with me for a minute. Oh, did it not? I think because I closed out, it might have not transferred those notebooks. So give me a second to figure out where I saved them. Let me check. Okay, it doesn't like that I have multiple windows up. Let me try this one more time. I had a backup analysis. When you do quite a bit of these webinars, oh, that's still not the correct one. Let's see. I think I have a few too many of these Jupyter notebooks launched. Let me try one prime. Okay, so this refreshed. So I'm going to go back to an instance that was already running. So I'm going to click on that link. Go to, which one is this? I think it should. No. That one. All right, so that plan backfired of having multiple Jupyter labs running. That's still not the run. Okay. So I'm going to get these outputs just to show really quick of 0, 1. That's not going to work. I can copy this here. So right now I'm saying I'm going to get this from my notebook. I wonder if it's going to go. Okay. And then I wanted to get the other notebook. So these are two notebooks. I'm not sure why they weren't uploaded into the shared folder. Oops. But we'll go with them. All right, so you see that I'm transferring from the data star here, and they appear on the left. So these are two notebooks if you want to get acquainted to what Jupyter Notebook is. So these are instructional notebooks that I've maintained and sort of developed, adapted from other folks. So one good practice is I leave information on, make a copy, and then begin working in that notebook so that you always have something to build upon. And if you haven't seen a Jupyter Notebook, this is more of a sort of instruction built-in text, but with code cells that you can execute. So for this, I'm going to go ahead and restart and clear outputs. And you can double click on these boxes and it'll edit and change that. You can say, all right, I'm going to enter my name here, and then I'm going to add in today's date of this. And if you hold down Shift Enter, it'll return it back. So it's a way that you can update and modify and keep different things in this interactive computing environment. One thing to notice is that you always want to think of a record of computation. So like a wet lab notebook you have in your physical lab space. What do you do for all the steps when you're running your bioinformatics analysis? So some of the things that you want to keep aware of is the name of the notebook you're working in. You can come to the left sidebar and rename it if you want. So these, it'll take time to get used to. There are different icons that you can practice getting used to. Again, this is already pre-put together, but what are we looking at? So how this is presented is through what we call Markdown. So if you click on this cell, this is where you have your editing text, and you have this pull-down number or menu that says Markdown. If you look at this code cell below, you'll notice it has these brackets, and you can type in code, and I'm going to hit Shift Enter to run it, and this will list what contents are in my current working directory. And you can see that the notebook, the other notebook I transferred, and the Vice in work. If I tried to type in list and Vice and reran that, you'll see that my gut microbiome folder is there. So these are ways that you can interact between the two. And if you want to learn about adding cells or even cutting cells, I built in some exercises that if you want to get used to all of this, you can work in that notebook. So these are building blocks because the next notebook is quick Unix commands that you'll need in order to run QIIME 2. One thing that you'll notice in these notebooks is that they don't have prompts. So these code cells are, in a sense, if you used a terminal, where you would go ahead and put your instructions that you want the computer to run. So if you're new to learning about your file system, this says type pwd. Again, this is a code cell, so you press Shift Enter. Getting used to these shorthand commands of ls that are list. This is going to, again, show what's in our current working directory. You can get used to a little bit of the flags here. So this will also list the contents, but in a long human read form. So these are ways to help you get used to understanding what's in your file system. So the other thing is to make a new directory. Spaces are important. So that's one thing I would encourage you to think about when you're learning about this setup. So, okay, I'm gonna do one last thing and see if I can find, I don't know if I have the last, no, okay. I'm gonna check one last time into the community data of where my folder that I thought I shared is housed, because I don't think I saved it. So let's go back to collaborations, example data, and see if I can upload that one last time. It might have just not copied. Okay, oh, there it is. So let's see. I'm gonna copy this really quick. let's see if that works. I get Control A. Oh, I need to, oops, say that this is the F. I can't remember the name. FF demo. Sometimes I regret naming them this way. So 03, because I don't think I can use autofill or autocomplete in there. So bear with me for just a few seconds. I'll show you demo QIIME. I'll be able to show you the Jupyter notebook if I type that correctly. QIIME. Okay. Yeah, okay. So this is, again, just a demo. I'm hoping to finish this for the whole QIIME 2 workflow. So again, I tried to keep the same idea that make a copy just in case you accidentally delete some of these code cells. But this notebook analyzes a gut microbiome sample. Again, these notebooks are great because you can put in links that go directly to the article that you want to reference, where the data are. And going back to this overview of this notebook is made for the Vice discovery environment. And I also want you to know that all of these are bash environments for the different notebooks. So we're not moving into Python or R just yet because you don't really need to run Python. You just need to execute the scripts for QIIME 2. So we already have our sample data set downloaded from the data store using iCommands. You can do that, but this, since it's already in Vice, that's one thing we want to keep in update. I'm going to change into Vice right now and run these cells, and then I'll make a new directory called gut microbiome outputs. And you can see that this is a way to navigate through, and you can leave updates. So I'm not doing any modifications to the text right now, just because I want to get through this. And to check out what types of plugins you have accessible for QIIME, you run QIIME info, and that should be able to launch QIIME to run within the Jupyter notebook. I added in, just what a plugin is as when you're reading through and understanding a few things. So since this is version 2018.11, all the available plugins from QIIME are listed here. So if you want to import data, I'm going to go through and demo that so you can see that this runs. So to import data, you have a type. So QIIME has this input of sample data paired in sequences with quality. So when I look at the gut microbiome, yeah, this is one. All these FASTQ files, they are the reverse and forward and reverse reads. And my input path is going to be that folder, the gut microbiome they're in. I know it's probably not the best naming, but in order to import for QIIME specifically, you have your input format. That's the Cassava 1/8 single lane per sample format. And you have to put in the tag of output file. So I'm going to put these in gut microbiome outputs because I just need that file. And you want to keep those differentiated from one another. And these backslashes, it's just nice to view what you're running, and if you need to fill in anything similar or different, that's one thing you should take into account. So whenever you're running a job, some of them ran quicker, so you wouldn't see the star, but the star shows you when you're running things. You can also go back to the terminal and type in top and see that, oh, QIIME is running here. When you're doing the declustering or denoising, I clicked Q to exit. You'll find that you'll see QIIME or R running depending whichever thing you need. So the import is going to take just a little bit, and if I go to my output file that I generated, you'll see that this .qza, the QIIME artifact file, is generated, and I can also visualize it in the sidebar to the left. Because we're getting close and I want to answer some questions, you can follow this. The paper sort of describes how many samples were put into this. the reads needed to be joined the reverse and forward. So in order to do that, you would use the QIIME vsearch join pairs. And to view a visualization file, that would be this .qzb. So I just want to show you that you can download this. And one last thing. So I'm going to go to right-click on that file we just generated. It'll go to My Downloads. I'm going to open up a QIIME2 view window. And you can see that drag and drop. So it might take a while to download. Once that finishes, you can drag and drop, and then I'll show you the province really quick. But one last thing, you know, this is a very short demo because I wanted to make sure this worked for the Vice, but you can also use commands within here to transfer all the outputs back. So if I put output, run that. Okay, we'll come back to that in a second. Let's go back to QIIME2 Viewer since the file's done downloading. And here, since it's a QZA artifact, it's not a visualization file, you'll just get this input or information about the name. If you go to Provenance and you click on that box, you'll see the action details, all the parameters set, and all the input files that are here. So at every step that you're running QIIME2, you'll get more input out. So I'm about 10 minutes over, so I'm going to stop there. And if you have any questions, you feel free to email me more if you have to head out. But I can go ahead and answer some questions now.
Thanks, Jocelyn. I don't see any questions in the chat window. Might take people a minute to think of some, or if something in terms of what you've presented. So maybe in this last couple of minutes, you can explain to people some of the, like, review just a few key steps, or how to get to your demo so that they can test on their own and replay it.
Okay, yeah. So there was a bit of a mix-up because I had multiple instances up, and I thought I had some of these files running. But just to go back to the live demonstration for a quick launch, I'll open this in a new window. You can take that link and go to the list of Vice apps in DE. You can see here the Jupyter Lab. Those will go over the steps on how to launch, but if you want to just click on the window, that'll open and prompt you to sign into CyVerse. Since I'm already logged in, it's going to bring in all the items, but the main thing you will need to fill out is the app information. So this is, again, what input files do you want to add in, and if you have your own data, you can upload using CyberDuck and access those from the data store. So that's one way that you can get back to the app from here. Once that pops up, you'll have this bell to the side that'll notify you that after you've launched an analysis, you'll say access your running analysis here. That link is what takes you to your Jupyter Lab. So I think if I open this one, it's the one I just real-time launched earlier. So this won't have any of the, I think it'll just have the gut microbiome. Let's see. No, so it has all the files I wanted in the last demo. Okay, so if you wanted to reproduce what I did and selected that QIIME2 file, you'll get the raw reads from the paper. You'll get access to the 3 Jupyter notebooks so you can get familiar. Whenever you're going through some of the downstream QIIME2 analysis, you'll need to have your metadata. The individuals who published this paper, they did a really great job. All I had to add in was this row of identifying the QIIME types of categorical or numerical. So I did more of the components. So if you go back to the journal article and look at data availability, what I did was download from the European Nucleotide Archives, get the raw reads, rename them for their sample ID. but the metadata and all the other information are on this paper. So I really liked how this was a public data set that I could share, because I couldn't really share with you if you wanted to reproduce some of my unpublished data. It'd be kind of tricky. So this is a great way to start, and if you wanted to go from raw reads, I can also help you with that if you want to get better at downloading public data sets at another time.
Jocelyn?
Yes.
There is a question about someone trying to upload data, the sequence data, but when they tried to upload, there was a continual question or an ask for metadata files. Can you explain that part a little more?
Within QIIME? Okay. Let's see. So I'm going to go ahead and type in. So I don't know if the user, they had an issue with the import, or if it's the next step of denoising or the clustering step.
If the person who wrote the question Christopher Cullis, could clarify that. I think you had said in your demonstration here that you needed to have the metadata file already in there. Does that?
Oh, so sorry about that. So the metadata comes into, well, it also depends on your sequence reads. So because this data set, the adapters were removed, so the metadata file for QIIME2, you would either need to put that under a barcode column, and that, if we go to QIIME2, they have documentation. So QIIME2.org, you go to documents, and one of the areas of importing data under their tutorials, you'll have to talk with your sequencing center what type of sequence data you have. The one that comes from this Frontiers paper is the forward and reverse feed reads. So if you're on older sequencing instrumentations, you might have just one FASTQ file and another that contains barcodes. So that's, again, one of the biggest barriers of QIIME is getting your data imported initially. They've gotten to fill out their documentation that assists with FASTQ data from the Earth Microbiome Protocol or the Cassava 1.8. So those are some of the more trickier areas that you have to specify. When you import, you may need the metadata file ahead of time, depending if you have those reads, I believe. But if you're using just the demultiplex, so that's the removal of your barcodes and adapters, and you're just importing the sequences, then you don't need any metadata file associated to that. So those are the differences when you're importing data. And if you have older data, you're going to have to use manifest files. So this is probably from 2016, 2017 sequence data. So that you might have to go into the forum to ask more questions for this. So I see some folks in the chat.
Okay. And to respond to another question, we will provide access to this presentation afterwards. We post everything, including the slides, to a wiki page, and we'll send you that link a little bit later, like I said, along with the YouTube video link, as well as a survey to get your input. So are there any more questions for Jocelyn while we're here? Okay. Jocelyn, thank you so very much. Really appreciate you presenting.
Okay, yeah. Okay, so I'll answer the one about data too really quick. So there are two ways in order to denoise and cluster your sequence data. So deblur does per sample, and it's a little bit quicker. For data two, when you're running that, it's looking at the whole sequencing run. So how it organizes and aligns all your sequences through all your samples. So that's going to take, depending on the number of reads in your sample sequence files, and then the number of samples you have. Some of mine that I've had, let's see, 50 samples that I'm analyzing. That's taken probably an overnight run for data 2, so it's about 16 hours. I've done smaller amounts on the Vice DE, but there is a limitation that we're trying to figure out how to add more memory, because that's one of the biggest steps for running data 2 is you run out of memory, and that's because you're constantly aligning and storing temporary clusters of your ASVs. So for that, I utilize Atmosphere's cloud computing, and we do have the QIIME software also running in Jupyter Lab on there. And I can share that with other folks if they need. It's on my GitHub repo for how you would go through and set that up. But yeah, so data 2 will take a lot longer. Blur is a little bit shorter because it's only doing per sample and not per sequence run. So,
thank you.
Yeah.
Okay, yes, somebody would like you to share your notes for sure.
Okay. Yeah, and I have other notebooks, so if you wanted to learn more, I can definitely connect. I don't know if my email's on anything. I don't think I got to that. I can add it in here if you want to shoot me leej2@hhmi.org. If you need any help, I can try to do my best to assist when you're trying to run on Atmosphere. So,
cool. Thank you. Okay, great. Well, thank you everyone for attending. Just a quick reminder that in 2 weeks we'll have the first of this 2-part webinar on nanopore sequencing analysis in CyVerse. And so with that, have a great weekend and we'll see you in a couple weeks. Thank you.
Yeah, thank you. Bye.